This lesson is about DNA mutations. Mutations are just a change of genetic material. Sometimes it happens at a chromosomal level. So there is one category, chromosome mutations. Sometimes it happens at a DNA level. So there's another category, a second category, DNA mutations, or mutations that happen in our DNA. Both of these things are random, but sometimes there are factors that might increase the chance of a mutation. For example, exposure to radiation or other chemicals. Internally, though, there are some things that can also contribute to mutations. For example, like a problem in cell division, like mitosis. A problem in mitosis could cause cancer. A problem in meiosis might cause something like Down syndrome. This diagram is sometimes referred to as the central dogma of biology. That is, the information in DNA is copied to RNA, which then is turned into a protein. Those proteins determine physical traits that we might have. So the code of DNA that gets copied to RNA is going to form or determine these traits that we have. If there's a problem in the copy of DNA, or if there's a problem with the RNA, that's going to affect the protein, that's going to affect a trait. Sometimes that effect can be devastating. Here are some examples of mutations in chromosomes. So the first example is a duplication mutation. This segment of a chromosome, maybe a gene on the chromosome, has been copied twice. Or perhaps it's a deletion mutation, where a segment of the chromosome is removed. Or an inversion mutation, where normally the gene sequence would go A, B, C, but this time it's flipped around. So it reads A, C, B. It just gets twisted. Sometimes there's a translocation mutation, where a segment from one chromosome is translocated. It's located to a different chromosome. On the DNA level, there are several different types of mutations. One type of mutation is a substitution. So we're just trading one nitrogen base for another. In the case here, a T is being switched for a C, and that's the mutation. Sometimes there's an addition mutation where we add an extra nitrogen base in, and then it's going to change everything after that. Or deletion mutation does the same. We remove one of the nitrogen bases, and everything after that is affected. Those are called frame shift mutations because everything after that mutation is interrupted. Take a look at these examples of mutations. Notice that each of the words only has 3 letters, and that's because codons are read in a set of 3. So our original DNA sentence reads, "The dog ate the cat," which kind of makes some sense. But take a look at mutation number 1. In this case, we've inserted a letter. By inserting that letter, everything after that gets messed up. In the case of mutation 2, we have substituted a letter. We've removed the T and substituted an R. You'll notice that it changes the entire meaning of the sentence. With mutation 3, we've deleted a mutation, and everything after that has been messed up. Sometimes when there are mutations, the result is silent. That is, these 3 nitrogen bases code for an amino acid. Sometimes, even though one of the nitrogen bases has been substituted, the code still reads the same. It still codes for the same amino acid. In that case, there has been a mutation, but the crisis has been averted because there's no overall effect. Sometimes when there's a mutation, there is called a missense mutation. That is, that one amino acid has been changed, and that messes up the protein. So for example, in sickle cell anemia, the protein that carries oxygen in your blood, hemoglobin, gets messed up, and it doesn't carry oxygen as well. And the blood cells don't maintain their shape very well, but they collapse into a kind of sickle-looking shape. Another example is a nonsense mutation. So in the case of cystic fibrosis, the protein here is messed up because everything after that in the protein sequence is gone. As a result, your body doesn't make a protein correctly, and you get like mucus buildup in your lungs, cystic fibrosis. An example of an insertion mutation is Huntington's disease. It's a brain disorder where, because of that protein's messed up, your brain cells deteriorate, usually starting around age 30 or 40. There's no known cure because it's the mutation in the DNA. Sometimes a deletion mutation might result in the loss of a protein. Depending on what that protein is, that'll determine how dramatic the effect is on the body. If that protein is super valuable, the effect is going to be dramatic. If that protein you can work around without it, then the effect will only be minor. Again, take a look at how these mutations impact the DNA, the RNA, and then the proteins. An example of a silent mutation, there's a mutation, but we still get the same amino acid. In a nonsense mutation, there's a mutation, and rather than continuing the protein, we put a stop in there that effectively stops the production of those amino acid chain, and the protein doesn't fold correctly. Or a missense mutation, where we have a totally different amino acid inserted into the chain, and sometimes that can cause some major problems. So I hope this helps you understand a little bit about the types of mutations and the effects that these mutations can have.